Hello student, welcome in the SIAM Genetic Engineering and its recent advancement course Regulation of Translation in Prokaryotes. Myself Dr. Tanushree Chatterjee from Raipur Institute of Technology welcome you all. So, first what is translation? Translation is the process of converting the information housed in mRNA into the protein sequence. Essentially, you are translating the language of nucleotides into the language of amino acids. The peptide formation is a dehydration reaction that combines the carboxylic acid of the upstream amino acid with the amine functional group of the downstream amino acid to form an amide linkage. Water is byproduct here. The ribosomes serve as the enzyme that mediates these reactions. It requires a mature mRNA to serve as the template and performs peptide bond synthesis in a directional fashion from the N to the C terminal of growing peptide or protein. This is known as N to C synthesis. To maintain proper protein function, the error rate of translation is approximately 10 raised to the power 4 or one error is every 10,000 amino acid encoded. The feasibility of protein synthesis is maintained by the ribosome's ability to match the code from the template mRNA strand with the appropriate amino acid. Template mRNA is read by the ribosome in groups of 3 nucleotides called a codon. The template is non-overlapping and read in discrete group of 3. This is known as the reading frame of the mRNA and it is always read from the 5 prime to 3 prime direction. Thus, for each mRNA there are 3 potential reading frames. Only one reading frame will be the correct one for protein synthesis. The ribosome must recognize and align the correct reading frame of the mRNA such that the correct codon sequences can be read. Small distinct tRNA nucleases, tRNA molecules are with specific amino acids and contain specific anticodon that complement mRNA codon sequences. Post transcriptional enzyme catalyzed modification of tRNA occurs at number of base and sugar positions and influences specific anticodon codon interaction and regulates translation, its efficiency and fidelity. This phenomena of nucleoside modification is most remarkable and result in a rich structural diversity of tRNA, of which over 93 modifications have been characterized. The variety of post transcriptional modification can be classified into 2 groups according to their complexity. The first group comprises the majority of modified bases which have simple methylations at the ribosome or base moiety that are usually introduced by a single enzymatic reaction. Simple modifications can be found at almost every position of the tRNA molecule with a high density in the tRNA core region, where tertiary interaction between D and T arm stabilize the 3-dimensional fold. The second group includes complex modification, whose synthesis require the sequential activity of several enzymes. Most often these hyper modified nucleosides are found in an anticodon of tRNAs, where they play a direct role in codon recognition and create what is known as vowel base and vowel position. The vowel base position is usually the first position of the anticodon read in the 5 prime to 3 prime direction, which aligns with the third position of the mRNA codon. This help to explain degeneracy found within the genetic code. Degeneracy means that a single tRNA is able to recognize multiple different codons within mRNA. Amino acyl tRNA synthesis. Amino acyl tRNA synthesis are universally distributed enzymes that catalyzes the esterification of a tRNA to its cognate amino acid. The product of this reaction, an amino acyl tRNA, is delivered by elongation factors to the ribosome to take part in protein synthesis. Amino acyl tRNA synthesis are named after the amino acyl tRNA product generated. As such, methionine tRNA synthase, that is charges tRNA met with methionine. The amino acyl tRNA synthesis catalyze a two-step reaction that leads to the esterification of an amino acid to the 3 prime end of a tRNA along with the hydrolysis of one molecule of ATP, yielding amino acyl tRNA, AMP and PPI. In the first step, term amino acid activation, both the amino acid and ATP bind to the catalytic site of the enzyme, triggering a nucleophilic attack of a alpha carboxylate oxygen of the amino acid to a alpha phosphate group of the ATP, condensing into amino acyl adenonylate, which remains bound to the enzyme, and PPI, which is expelled from the active site. The 23 known AARSS can be divided into two major classes based on the architecture of their active site, that is class 1 and class 2. In class 1 synthetase, the catalytic domain bears a dinucleotide or Rossmann fold, that is RF, featuring a 5 standard parallel beta sheet connected by alpha helices and is usually located at or near the N terminus of the protein. This RF contains the highly conserved motifs high and KMSKS separated by a connecting domain term connecting peptide 1, that is CP1. Class 2 active site architecture is organized as 7 standard beta sheets flanked by a alpha helices and features 3 motifs, which shows a lesser degree of conservation than those in class 1. If we see the ribosome structure, the ribosome is a highly conserved molecule machine in all organisms. It is composed of 2 unequal subunits which consist of a distinct set of ribosomal RNA, that is tRNA, and ribosomal protein RPS, that combines to form a large nucleoprotein complex. The ribosome structure in all living organisms harbor 3 different tRNA binding sites. The A site where decoding occurs and the correct amino acyl tRNA, that is AA tRNA, is selected on the basis of the mRNA codon displayed. The P site which carries the peptide tRNA and the E site which binds exclusively deacetylated tRNAs that are existing the ribosome. Thus, during translation the tRNA moves from the A site through the P and the E site where it leaves the ribosome. Prokaryotic ribosome have a mass of unit 2500 kDa and a size of 17 S units. Svedberg unit is a measurement or measure of a sedimentation rate in centrifuges and thus is representative of size. A complete ribosome, that is 70 S, can be dissociated into large subunit, that is 50 S, and a small subunit, that is 30 S. You can see in this picture. The small subunit is formed by the interaction of 21 different proteins and a 16 sRNA molecule, whereas the large subunit contains 34 different proteins and 2 RNA molecules are 23 S and a 5 S species. Now, initiation of protein translation. The small subunit of ribosome, that is 30 S, interprets the genetic information by selecting amino acyl tRNAs cognate to the mRNA codon in the decoding center. The large subunit 50 S carries the catalytic peptidyl transferase center where amino acids are polymerized into a protein. A small and a large subunit unite together at the start codon of a gene to form the 70 S ribosome and dissociate again at the stop codon upon completing the synthesis of the encoded protein. The initiation phase of protein synthesis involves a limited number of actors aside from the 2 ribosomal subunits. Key roles are played by the initiators tRNA, the translation initiation region, that is TIR of the mRNA, and 3 protein factors. The initiation factor IFS, IF1, IF2 and IF3 that ensure speed and accuracy to the overall process. The initiation tRNA contains a methionine residues that has been enzymatically modified to contain an N-terminal formyl group. T-met is only used for the initiation for protein synthesis and is thus found only at the T-terminus of the protein. Unmodified methionine is used during the rest translation. Once protein synthesis is completed, the formyl group or methionine may be removed by peptide deformylase, and on occasion the entire methionine residue can be further removed by the enzyme methionine aminopeptidase. The TIR sequence with the mRNA contains the start codon and usually an upstream untranslated region that interacts with the small subunit of ribosome. The bacterial cell produces expresses a plethora of different mRNAs with different TIR sequences and structures. The efficiency by which these individual transcripts are translated depends not only upon the abundance and stability, but also upon the nature of TIR. Thus, unlike the other aforementioned actors that represent constant, the mRNA TIRs represent essentially the only variable in the process of mRNA initiation, site selection and can affect translation efficiency. Although the triplet AUG is by far the most frequent initiation codon found in TIRs, other initiation triplets are found in bacteria and the central U is the only universally conserved base of start codon. Among the triplet, those having a 3 base G are recognized equivalently and most efficiently by IRF3 during the initiation complex formation. Another important characteristics of a large number of bacterial mRNA TIRs is presence of Shine-Dargano or SD sequence that is complementary to the 3' end sequence of 16S tRNA, the anti-SD sequence or a SD. The SD sequence when present is usually at optional distance of 4 to 9 nucleotides upstream of the initiation codon, while the SD sequence plays an important role in the efficient translation of many mRNA transcripts. It is not essential. The 3 protein initiation factors IF1, IF2 and IF3 determine the kinetics and the fidelity of the overall initiation process. The 3 IFs are bound 1 copy each to a specific site of the 30S subunit, where they assist with the formation of the initiation complex and assembly of the 17S ribosome. The initiator tRNA is first amino acylated with methionine whose alpha NH3 group is eventually blocked by a specific formyl transferase TMF to produce a tRNA molecule. This modification prevents interaction with the elongation factor EFTU. Blocking EFTU binding ensure instead the recognition and binding of tRNA by initiation factor IF2, effectively docking it with the 30S subunit. tRNA binds with high affinity to the ribosomal P site, unlike all other amino acyl tRNAs that bind to A site in a ternary complex with EFTU and GTP. To form the 30S initiation complex, IF3 and IF2 are the first factor to bind to the 30S subunit, forming an unstable 30S IF3 IF2 complex. The binding of IF1 causes a conformational changes in the 30S subunit, stabilizing the complex and allowing the recruitment of tRNAs by IF2. IF1 binds in a site of the 30S subunit, where it contacts ribosomal protein S12. Recruitment of the tRNA can also stabilize the mRNA interaction with the 30S subunit through the formation of hydrogen bond between the codon of the mRNA and the anticodon of the tRNAs. The recruitment of the mRNA and the tRNA to the 30S initiation complex loaded with the IF2, IF3 and IF1 initiation factor, the 50S subunit is very rapidly docked to yield an initially unstable 70S initiation complex. The IF2 protein is a GTP hydrolase enzyme and as such binds with the cofactor GTP prior to the recruitment of the 50S subunit. Contact between the IF2 GTPase activation center with the 50S subunit causes the rapid hydrolysis of GTP to GDP and PI. The formation of the 70S complex causes the dissociation of the initiation factor. IF2 is the last factor to be dissociated, leaving the ribosome after having positioned tRNA in the P site of the 70S initiation complex. It must be placed in the correct orientation to facilitate peptide bond formation. GDB and PI also dissociated from the complex with the removal of IF2. The elongation factor EFG is then free to chaperone with the first tRNA into the A site and the first peptide bond is formed. This marks the beginning of the elongation phase of protein synthesis, the elongation phase of the translation. The prokaryotic elongation phase of transcription require the activity of 3 primary elongation factor, that is EFTU, EFTS and EFG. During elongation, amino acyl tRNAs are delivered to the ribosome in the form of ternary complex. The tRNA are translational GTPase and the GTP molecule, the tRNA decodes the information on the mRNA by forming hydrogen bonds, that is H bond hydrogen bond and between codon and anticodon nucleobases. Remarkably, the free energy difference correct, that is cognate, and incorporate incorrect base pairing alone does not explain the very high fidelity of decoding. High fidelity is achieved by a 2-step decoding process: initial selection leading to GTPase activation and pro-threading. In addition, the free energy difference. Kinetic effects contribute to the discrimination. The GTP hydrolysis rate is increased and tRNA rejection rate is decreased by the recognition of the correct codon. Small unit nucleotides A1492 and A1493 adopt a flipped out conformation in the presence of a tRNA, and this conformation, the tRNA anticodon hydrogen bonds with the codon of the mRNA forming a mini helix structure. The flipped out nucleotides A1492, A1493 along with G530 were found to shield the codon anticodon base pairs from solvent. This shielding of near cognate base pairs from the solvent is incomplete, causing an increase in the free energy difference between near cognate and cognate base pairs and more flexibility within the docking region. This reduces the strength of hydrogen bonding between a non-cognate tRNA and causes the inappropriate tRNA to leave the A site before peptide bond formation can occur. This increases the fidelity and the discrimination of tRNA selection such that only the correct cognate tRNA is incorporated into the A site. After GTP hydrolysis, the GTPase EFTU dissociated from a tRNA. At this point, the EFTU is tightly bound with a molecule of GDP and cannot release GDP on its own to be recycled for a second round of tRNA. The recharging of EFTU is executed by the elongation factor thermostable. The binding of EFTS with EFTU GDP causes a conformational change in EFTU that allows the release of GDP. The binding of a new molecule of GTP with the EFTU protein causes the dissociation of EFTS and fully recharge EFTU. Dissociation of EFTU from the ribosome allows the tRNA to move into the peptide transferase center on the large subunit. At the core of ribosomal translation is the catalysis of peptide bond formation. The current reaction model point to a substrate assisted mechanism. Once the peptide bond is formed, the ribosome needs to translocate down the mRNA to make the next mRNA codon available within the A site. This also requires the shifting of tRNA molecule such that the tRNA in the A site, which is now tethered to the nascent peptide, shift to the P site. The P site tRNA, which is now empty, shifts to the E site, and if there was an empty tRNA in the E site, it will shift to the exit the ribosome. Shifting the tRNAs and mRNAs within the ribosome core requires the action of the EFG elongation factor. EFG is a GTP hydrolase protein that binds to a site of the ribosome. The EFG protein has high flexibility that enables it to act as a hinge. Folding of EFG is dependent on GTP hydrolysis. Thus, when binding to the ribosome, the fast hydrolysis of GTP act as a power stroke, folding the EFG protein and causing a conformation shift in the ribosome that enables translocation of the tRNA residues and the mRNA. Translocation of tRNAs is accompanied by a large scale collective motions of the ribosome, relative rotation of ribosomal subunits and L1 stalk motion. L1 stalk, which is a flexible part of the large subunit, is a contact and moves along with the tRNA from the P to E site. Once in the EFG GDP form, the factor quickly dissociates from the ribosome, opening up the A site for the recruitment of the next AA tRNA molecule. The elongation cycle will continue to the repeatedly until a termination codon is reached. Now, translation termination. Termination of bacterial protein synthesis occurs when a stop codon is presented in the ribosomal A site and is recognized by a class 1 release factor, that is RF1 or RF2. These release factors have different but overlapping specifications, where RF1 reads UAA and UAG and RF2 reads UAA and UGA with strong discrimination against sense codon. The RFS are multi-domain protein where binding and stop codon recognition by domain 2 at the decoding site cause the universally conserved GGQ motif of domain 3 to insert into the A site of PTC some 80 S away from the decoding site. This event triggers hydrolysis of the peptide tRNA bond in the P site of the PTC, and the nascent peptide chain can then be released via the ribosomal exit tunnel. After peptide release, F1 and RF2 dissociate from the post termination complex. The dissociation is accelerated by class 2 release factor called RF3, which function as a translation GTPase that binds and hydrolyzes GTP in the course of termination. While RF3 increases the efficiency of peptide hydrolysis, it is not an essential protein for the process. In gene knockout studies, RF3 is dispensable for the growth of E. coli and its expression is not conserved in all bacterial lineage. For example, RF3 is not present in the thermophilic model organism of the Thermus and Thermatoga genera and in Facetious, in Chlamydelis and Spirochete. This means that both RF1 and RF2 are capable of performing a complete round of termination independently of RF3 or that other GPTAs from the elongation or initiation phase of translation can compensate for the action RF3. The release factor RF1 and RF2 acquire an open conformation on the 17S ribosome, which is distinctly different from the closed conformation observed in crystal structure of free RFS. The conformational equilibrium of the free RFS in solution shows that this open conformation is dominating at about 80%. During peptide hydrolysis, the RF factor cause rotational and conformational changes within the ribosome and that allows the binding of a ribosome recycling factors and the EFG GTPase, which leads to the dissociation of the large subunit from the small subunit and the release of the mRNA. Thank you all.